Hello, everyone. I would like to welcome you to today's webinar, Unraveling the Dual Role of HNF1 Variants from Rare Diabetes to Atherogenic Lipoproteins. My name is Lucy Sue. I'm a Senior Product Manager at Twist Bioscience. Before we start, I'd like to remind you of a few housekeeping items. I would like to give you a few housekeeping guidelines. So all lines will be muted during the webinar. If you have a question, please submit it in the Q&A box rather than the chat box, and we will answer all questions after the presentation. If you share your name, we will be able to get back to you after the webinar if your questions have not been answered. Following the presentation, if you have 1 minute to take a brief survey, we would appreciate your feedback so we can continue to improve. The survey will open automatically in your web browser. I'm very excited to introduce you to our speaker for today, Amit R. Majithia, MD. He's an associate professor in the Division of Endocrinology in the Department of Medicine and at the UCSD School of Medicine. Dr. Majithia received his bachelor's degree in chemistry and physics from Harvard University and his MD from the NYU School of Medicine. He's a clinically active endocrinologist with a lifelong passion for improving the life and health of people with diabetes and cardiometabolic disease. Dr. Majithia is an accomplished educator and researcher, serving as a member of the teaching faculty in the Biomedical Sciences graduate program and the Bioinformatics and Systems Biology program, the Genetics Training program, and as an affiliate with the Division of Biomedical Informatics at UC San Diego. His research focuses on using the tools of functional genomics, bioinformatics, and human genetics in a collaborative, multidisciplinary research environment to advance our understanding of diabetes and cardiometabolic diseases. The Majithia Lab participates in several research centers, including the UCSD, UCLA Diabetes Research Center, Altman Clinical and Translational Research Institute, and the Institute for Genomic Medicine, as well as the Center for Engineering and Diabetes in the Institute of Engineering and Medicine. Through these research initiatives, Dr. Majithia is dedicated to improving the diagnosis, treatment, and management of diabetes and cardiometabolic diseases, ultimately improving the quality of life for patients affected by these conditions. And with that, I'll hand it off to Dr. Majithia.
Thank you, Lucy, for that very kind and generous introduction. And thank you to the team at Twist for giving us the opportunity to share our research with this group, and thank you to the audience for attending in what might be a bit of an early morning. So I'm going to turn my camera off because of bandwidth issues as I go through our research. And today I'm going to talk about a project that we did using deep mutational scanning to identify human gain-of-function variants in a protein HNF1 alpha that's very important to diabetes. I'm going to start with a background and a paradigm of what we're interested in, because the techniques that we used and developed here are applicable beyond diabetes, beyond HNF1 alpha, and I think this is a really exciting area of research that can make progress in multiple genes and diseases. So as Lucy mentioned, my lab works on the translational investigation of insulin resistance. So just to say a couple of words about that, you might have heard of insulin resistance as prediabetes or obesity. They're sometimes used as synonyms, and insulin resistance is the underlying physiological state that occurs when someone carries excess weight on their body, excess adiposity, or they have prediabetes. And this is really an ongoing pandemic in our society and across the world. So it's estimated that 90 million people in the United States have prediabetes, and over 90% of them don't even know it. So new therapies and new tools to both diagnose and treat insulin resistance related disorders like diabetes, fatty liver, and cardiovascular disease are really needed. And so that's the focus of our lab. And our approach combines what I'm going to call experiments of man, experiments of nature, and experiments on man. And so To break that down a little bit, the experiments of man are what we do in the lab. And so in our lab, we do CRISPR screens and deep mutational scans. And today we're going to talk about deep mutational scans. Experiments of nature, which really boils down to human genetics. And over the past decade, these wonderful resources have been made available through both the generosity of governments as well as people. And so we participate in a number of biobanks, including the UK Biobank, the Million Veterans Program in the US, and the All of Us Research Program in the US. And this is genetic research that has been empowered by people really donating their data to science. And I'll talk a little bit more about that. And then finally, experiments on man. And we're talking about clinical trials, both that have been done in the past, like the diabetes prevention program, and clinical trials that we have underway in my lab. So one of the fundamental goals that we're trying to approach is how do we find genes that have therapeutic impact? How can we know that if we design a drug against a gene, and there are many ways to design drugs now, not just small molecules, but antisense oligos, but how do we know actually doing that thing to the gene is going to result in a benefit to human health? So what we want to know is the mechanism. How does it work biologically? The direction of effect. How does moving the gene function up and down relate to what we care about ultimately, which is the disease? And then we want to know if it actually works in people and what are the side effects, because every drug and every gene, when you modulate it, has effects that you want and potential side effects as well. And so the opportunity, this activity has been done for almost a century in all drug development, but the opportunity today is to leverage nature's clinical trials, which is human genetics, through biobanks, and I'll talk about that a little bit more. The challenge is now functionally characterizing variants, and that's where the deep mutational scanning is going to fit in. And so what we want to define, taking a human genetic approach for gene function to validate a gene as a potential target for improving human health, in our case with regard to insulin resistance, is to define an allelic series. And that means over here on this graph, that if you have different mutations and they affect the gene in different ways, they cause the gene to decrease its function or increase its function, then the biological phenotype, this could be insulin resistance, cardiovascular disease, cholesterol levels, anything, there has to be this kind of positive relationship. It could be an inverse relationship or it could be a positive relationship, but there has to be a relationship. And that tells you that actually creating a drug against this could possibly work. If the relationship is like in blue, then that's not really a good target. And so what we want to know, using these allelic series with your different genetic variants that cause a different change in gene function, is the efficacy, does it work, the safety, people who inherit these mutations, are they otherwise okay, and establish the mechanism of action. And just to build upon that a little bit, what we want to know, if you do have a relationship between the dosage, the alleles, and the gene function and the phenotype, there is the therapeutic window, because all genes and all drugs have side effects. And so you want to know that if you move the function up or down and you're getting the response that you want, is the response of something you don't want very different. If these two curves are very close to each other, then there's no way to activate or inactivate that gene without causing both the beneficial and negative consequences. And so that's the idea of therapeutic window, and genetics can help us do that as well. So that's a little bit theoretical, and I want to make it concrete by talking about what I think is one of the most inspiring examples in human genetics of identifying a target and then using it to create a drug that actually improves people's health. And so here, this is PCSK9, which is a gene that's expressed in the liver and that participates in LDL receptor recycling. And it was found well over a decade ago that individuals who had this particular variant, so at the 46th amino acid, they had an L, um, uh, had decreased cholesterol, so significantly decreased cholesterol, and they were protected from heart attacks. So they had half the hazard for heart attacks. And this was proved in multiple studies, and so it was through functional investigation, it was found that that L actually decreased the function of the gene. And so this is the idea that we could inhibit the gene function and potentially get a benefit on people's cholesterol and potentially a benefit on heart attack. And ultimately, the mechanism was worked out that this gene encodes a protein that prevents LDL receptors from degrading. The LDL receptors are expressed on the liver, and they take up the cholesterol from the blood. And so instead of essentially clogging up the coronary arteries and causing heart attacks and stroke, it gets metabolized by the liver. And so an antibody was created, and now there are siRNAs on the market as well, to block PCSK9, increase the level of LDL receptors, and then decrease LDL. And this is some clinical data on 35,000 patients who are already taking the best cholesterol medications available on the market. And this is statins, which have saved millions of lives. So these people are already on statins. Their cholesterol levels start off at 90. I wish I had a cholesterol level of 90. That's already very protected. And on the PCSK9 inhibitor, the levels went down to 30. So this is an unprecedented degree of effect. And it was shown that people who had natural disruptive mutations in this gene, so they were missing PCSK9, were otherwise totally fine. They just had lower cholesterol and lower heart attacks. And so this has become an incredibly successful part of our armamentarium to treat lipids and cardiovascular risk. So what we hope to do by human genetics is to actually shortcut some of the incredibly laborious and expensive process to identifying and validating a target. So traditionally, if you have a hypothesis that a gene from a knockout study in a mouse or in cells is going to do something beneficial in humans, you would have to create a molecule. You'd have to identify the gene. You have to validate it by knocking it out. You'd have to create a molecule that replicates those properties, and then eventually get it into people. And so this is overall a 10 to 15-year process. And most of the time, the process fails not because there was some kind of toxicity, but actually 51% of late stage failures in drugs are due to lack of efficacy. So that initial hypothesis from 15 years ago that the gene actually did something was incorrect. It didn't work in people. And that's where I think human genetics can really help by de-risking that and giving you information in the way those people with PCSK9 mutations de-risk the process of development for PCSK9. Now, I presented that one variant that we knew the function of in PCSK9, and that's over here. So that variant decreases the function of PCSK9 and decreases LDL cholesterol. But if you look over thousands of people, there are many, many different protein coding PCSK9 variants. And some of them you can interpret because, look, these are nulls. And so human knockouts, essentially, and this one's a human knockout, and you have a decrease. Some of them actually increase PCSK9 function, and that increases LDL cholesterol. So it all fits together. But if I took away these colors, you can see that the dots are kind of all over the place. So there's no way to actually find the signal in the noise. It all looks like essentially random because most variants have no change in function. Some of them decrease function, some of them increase function. So understanding the function is key. And what I want to propose is that we use deep mutational scanning to do that. So to introduce the idea of deep mutational scanning, this is not a new concept. It's essentially a synonym for saturation mutagenesis, but the technology has evolved where we can approach entirely different kinds of genes and mammalian mammalian cells using these methods. So this was a study I did during my postdoc, which was one of the first modern deep mutational scans. And we studied the gene PPR gamma, which is a transcription factor, a master regulator of adipocyte differentiation, and one of the only targets for insulin resistance for a class of drugs called thiazolidine diones, which are the only class on the market that directly targets insulin resistance. So we essentially, this was in the pre-twist era, synthesized every single amino acid permutation of PPR gamma, put them into cells where we knocked out PPR gamma, and then looked at, by flow cytometry, the PPR gamma function with respect to CD36, which is a cell surface marker and a direct transcriptional target of PPR gamma. Then we sorted the cells according to the CD36 high and low population and did shotgun sequencing to recover the variant identities in the low CD36 pool and the high CD36 pool, and then we were able to compute a score for each of them. And this is how we created essentially a lookup table for what every single amino acid change in every position of PPR gamma does to the protein. And I want to say my partner in this, Tarjay Mickelson, who has since moved to industry, he created this very robust method to assemble oligos into these transgene libraries that contain every amino acid variant. And the leap there was that instead of having 30% coverage of the variants, we had 99% coverage of the variants. They were very balanced. And in the next study, I'll show you that we no longer synthesize those libraries ourselves because it was incredibly time-consuming and laborious and requires a person working for essentially half a year or a year, and move to twist. And I'll draw some comparisons. So, and a little bit of background about biobanks, because we're going to be using this data to make our inferences, to identify variants, and then make inferences to what they do in humans. So this is the UK Biobank. So it's 500,000 people from the UK, so mostly of Northern European ancestry, who have, in an act of incredible generosity, donated all of their medical records, undergone imaging tests like MRIs, filled out hundreds of surveys, their blood tests, everything, donated that to science, and they have been fully exome and now are going to be whole genome sequenced. So we know all of their variants, and we know all of the things that happen to them. And this is available to any investigator across in the world. And so the principle here is that from exome sequencing, we can identify all of the naturally occurring variants that are protein coding. We can use a deep mutational scan and hone in on a gene of interest and interpret the function of those variants. And then we relate the function to the patients, their actual phenotypes that are recorded in their health records. And so Natalie DeForest, a graduate student in my lab, who led the HNF1 alpha study, before that, she used this deep mutational scan that we'd conducted for PPR gamma, identified all of the protein coding variants in PPR gamma in the UK Biobank. That was 1 out of 500 people contained a rare protein coding variant, used the deep mutational scan data to interpret it, and then looked at what are the clinical effects of increasing and decreasing PPR gamma function through naturally occurring protein coding mutations. And remember, this is a positive control because we already have a PPR gamma activating drug that's been used to treat hundreds of thousands of people. And what we recovered was that if you increase PPR gamma function, you increase body fat. That's a known side effect of the thiazolidine diones that activate PPR gamma. You do decreased waist-hip ratio, which is a measure of central adiposity. PPR gamma is known to do that. And you do decrease insulin resistance, and that's measured by the triglyceride over HDL ratio here. And that's a known effect of PPAR gamma agonists as insulin sensitizers. You also decrease liver enzyme levels, and thiazolidinediones are one of the only drugs, except in the past year, that's been known to be able to treat fatty liver disease. And you actually decrease hemoglobin concentration. And so it's known that one of the side effects of thiazolidinediones is to increase fluid retention and dilute your blood. And so we recovered all of that. And so then we moved on to HNF1 alpha. And this is a paper was recently published. Natalie led this work. And so to give you a little bit of background about HNF1 alpha and the reason we were interested in it is that this gene has been known for over 20 years as a monogenic cause of diabetes. Monogenic meaning that a single disruption in this single gene is enough to give you diabetes. And it's a syndrome of diabetes called MODY, maturity onset diabetes of the young. And so it's an autosomal dominantly inherited syndrome. So that occurs in 1 out of 100,000, 1 out of 250,000 people that have MODY3. And it was noted over many decades that these individuals also carried altered levels of serum lipoproteins like LDL and HDL cholesterol. They had different levels of CRP in their blood. And there was a common SNP in HNF1 alpha, which was associated with not only type 2 diabetes, but with cardiovascular disease, but it was unknown how any of this worked. So what Natalie undertook to do was, in the UK Biobank, identified all the people who had protein coding variants in HNF1 alpha that were rare. And she identified 4,000 such individuals, and then performed a deep mutational scan in HNF1 alpha to interpret the function of those variants, and then related it back to what's happening to their risk for diabetes, what's happening to their blood glucose levels, their CRP levels, their lipid levels, etc. And so this was a library that Twist synthesized for us as one of their early stage testers, beta testers, perhaps you could call it. And the HNF1 alpha gene is actually a little bit longer than PPAR gamma, being 631 amino acids. So this library contained, the full library would have contained 11,970 variants. And the library that we received, we sequenced it to check for its balance and its comprehensiveness. And so it was over 99% comprehensive of the desired amino acid changes. And it was very balanced because you don't want one amino acid to dominate any other amino acids at any particular position. And so those are the two metrics by which you would judge a site saturation library like this. And so using a very similar conceptual method, this time we're using liver cells because HNF1 alpha is hepatocyte nuclear factor. We knocked out HNF1 alpha. We introduced the Twist HNF1 alpha saturation library. And now, instead of CD36, we're looking at TM4SF4. And HNF1 alpha is also a transcription factor and activates the cell surface expression of this gene. We knock out cells for HNF1 alpha have the red level of TM4SF4. Wild type cells have the green level. And then the library has a mixture of low functioning and high functioning variants. Again, we sorted them. We sequenced the variants that are embedded within the cells, count them up, and create an enrichment score. And so here is our lookup table for HNF1 alpha. What you can see in this heat map by color is that there are known DNA binding domains of HNF1 alpha, which are highly structurally conserved. And so most amino acid changes in those domains tend to break the function of HNF1 alpha. So, and here you can see the average function score per position. And so that's what you would expect, that if you take a highly conserved domain, you introduce mutations in it, most of those mutations are going to make the HNF1 alpha work more poorly. And this is a different representation of the data where we've taken all of the 11,970 variants, ranked them in order of function, So you get this curve, and the known variants that have been associated with MODY3 that have been discovered over 20 years, they have low function, as we would expect. So our data are good. Surprisingly, we found that about a third of the variants, sorry, not a third, but about a sixth of the variants, actually increase the function of HNF1 alpha in our assay. And so we wondered, does this have anything to do with people, or is this just an artifact of the way we did this experiment? And in fact, there happened to be quite a few individuals who carried gain of function variants in HNF1 alpha. So not only do we find it in our assay, but they're actually carried by humans. And so we selected a series of them across a range of the protein to study and validate whether they actually cause increased function, and what is the consequence in people. So even though HNF1 alpha has been sequenced for 20 years, people had focused only on loss of function variants. And so these were essentially hiding in plain sight. But because we had this comprehensive data and we're able to look at all variants together in context, it became immediately clear that there were these gain of function variants. And so using the gold standard assays for HNF1 alpha function, which are in these heterologous cell types like HeLa, on the albumin promoter, and then in insulin producing cell models, these are Min6, these are mouse cells, on the HNF4 alpha promoter. So these are have been established for decades. We find that actually, yes, compared to wild type, these variants have a tendency to varying degrees to increase the transcriptional activity of HNF1 alpha. And then we also saw that ANGPTL3, which is an emerging drug target for lipids, was increased. The secretion of it was increased by these gain of function variants in HNF1 alpha. So this wasn't just a cell line artifact. These variants are truly gain of function. Okay, so now what happens to the people who carry gain of function variants in HNF1 alpha? And this shows the data in two different ways. So we looked at the continuous allelic series of HNF1 alpha from loss of function to gain of function. And in fact, there is a decrease, a 30% decrease in type 2 diabetes risk in people who carry activating variants in HNF1 alpha. And so just to confirm that this wasn't all being driven by loss of function variants, we actually categorize the variants so into loss of function and gain of function. And the loss of function, as you expect from what we know about MODY, increases the risk of diabetes, whereas the gain of function variants actually decrease the risk of diabetes. So it's not being driven just by loss of function variants. And this held up in meta-analysis with an American cohort. Actually, this is an international cohort, which is the Accelerating Medicines Partnership Type 2 Diabetes Case Control Series, but it's much smaller than the UK Biobank, so you can see the error bars are wider. So then Natalie went on to look at what about the blood tests that these individuals had with these 4,000 people with protein coding variants in HNF1 alpha. And as you might expect, they had increased levels of C-reactive protein, which is a pro-inflammatory marker, because that's released from the liver and is driven by HNF1 alpha, and that's been known molecularly. They actually also had increased levels of LDL, a non-significant trend towards increase in triglycerides, and a decrease in HDL. So that's the good cholesterol, decrease in HDL levels. And meanwhile, they had a decrease in HbA1c, which is a measure of glucose, and that fits in with their decreased risk of diabetes. So HNF1 alpha is active in both the liver and in the beta cells of the islets, which are the insulin-producing cells. That's how it causes diabetes, by causing dysfunctional beta cells in islets. So Natalie used a different genetic instrument. So now she's using genetic variants called expressed quantitative trait loci. So these are SNPs that have the property that they increase or decrease gene expression in a particular tissue. So she selected genetic variants that would increase HNF1 alpha expression level in the liver only. And then she looked at, now these are common variants. So instead of 4,000 people, she had 400,000 people that she could look at this in. And found again that, yep, when you increase HNF1 alpha in the liver, you have increase in CRP. The LDL effect seems to be a liver-mediated effect. And the decrease in HDL seems to be a liver-mediated effect. But the decrease in glucose doesn't seem to be a liver-mediated effect. And so that fits in with what we know, that insulin is not produced by the liver, but by the islets, and that's what's controlling the glucose. So then moving forward a little bit and digging into the molecular mechanism, what Natalie identified was that if you knock out HNF1 alpha from liver cells, you get changes in a number of genes, including PCSK9 and ANGPTL3, this emerging factor. And that proved to be true across multiple hepatic cell lines. And then if you aggregate these genes into pathways, when you add back the gain of function variant as compared to wild type, the pathways that decrease, wound healing, complement, fatty acid metabolism, they seem to increase. So there is this reciprocal effect that knocking it out decreases these pathways, while putting in a variant that increases HNF1 alpha increases these pathways. And then she did these dose response curves by dosing in HNF1 alpha levels using some molecular techniques that I won't detail here. But if you dose in HNF1 alpha into cells in a dose response dependent fashion, you increase PCSK9, which probably explains the increase in LDL, and you decrease the levels of ApoA1, which is the major protein component of HDL cholesterol, and so that probably explains the decrease that we see in HDL cholesterol. So putting it all together, using this deep mutational scan to interpret the protein coding variants in HNF1 alpha, we have the following model: that HNF1 alpha gain of function variants protect you from diabetes, probably by increasing beta cell function and decreasing hemoglobin A1C levels, that's glucose, and that thereby decreasing your risk for type 2 diabetes. But these same variants in the liver have a negative metabolic effect because they increase PCSK9 levels, increase ANGPTL3 levels, and decrease ApoA1 levels, leading to increases in LDL cholesterol, a trend towards triglycerides, and decrease in HDL cholesterol. Ultimately, diabetes is a risk factor for heart disease, and so are these so is cholesterol levels, as you probably know, and these go in opposite effects. And we didn't have quite the power to test it, but we looked at cardiovascular disease rates in these gain of function variant carriers, and it essentially seems to wash out. So you have a bad risk factor going down and a bad risk factor going up, and we think that they seem to cancel each other out. Now, while our paper was in review, this interesting paper came out in Cell Metabolism, which worked out the mechanism of a phase 1 non-statin lipid-lowering compound. And what they found is that this compound, which decreases LDL cholesterol and triglyceride levels in the blood in people, worked by inhibiting HNF1 alpha dimerization, which led to decreased PCSK9 and decreased ANGPTL3. So essentially, through this chemical compound, they identified the same mechanism that we identified through genetics. So it was a nice proof of concept that you can find the same thing without creating, going through the trouble of creating a drug and treating patients with it. So to summarize some take-home points, population scale biobanks like the UK Biobanks contain these allelic series, these variants that change the function of genes in different ways across all genes. So whatever gene you're interested in, you can study that way. Um, but these variants are very rare. So you need to understand their function and you need to be able to put it together. You can't just look at one variant and draw a conclusion. And this idea of finding the variant's genotype, function, either through deep mutational scanning or another procedure, and relating it back to the phenotypes in the people, using these kinds of assays allows you to combine variant function to successfully credential whether a gene has therapeutic properties, what's its effect in humans, and what are potentially the side effects. And the deep mutational scans or saturation mutagenesis allows you to really turn that wheel very quickly instead of having to first find the mutations, then design them in the lab, and then do the testing. You essentially do all the experiments ahead of time. And with that, I want to thank you for listening. Thank my lab members, and Natalie's here in the middle. She led this study, a very talented person. And I want to thank my collaborators, Jerry Levsky, Jason Planik, Sven Hines, and Radha Venkatesan in India at the Madras Diabetes Research Foundation, and Gina Pelosa at BU. Thank you.
Well, thank you, Dr. Majithia, for a great presentation. It's always exciting to learn how our partners are leveraging our products. So now we're going to open it up for questions. If you have a question, please make sure to ask it in the Q&A box. So we have one question right now from an attendee. Their question is, could you please elaborate on how you sorted the cells using FACS or some other method? Thank you.
Yes, can you still hear me?
Yes, yes.
So that's exactly correct. In both the PPR gamma study and the HNF1 alpha study, we used FACS. There are different investigators have used different methodologies, and so when people are doing deep mutational scanning in the cancer field, for example, they use cell growth assays, and there you don't even have to sort anything. The variant that causes the cells to grow or not to grow will change its frequency just in the pool of cells. So that's an even easier experiment where there's no sorting. You just let the cells grow and then sequence the original pool and sequence what you get any number of days later. There are also strategies to use reporters, and so this rather than a cell surface marker, the variant can drive a genetically modified cell that produces a GFP or another color. So some investigators have used that as well. In this case, we're using FACS, and we're leveraging the natural response of these genes because we're studying transcription.
Thank you. So the discovery of these genetic variants with potential protective effects against type 2 diabetes and insights into their impacts on the hepatic physiology raises the questions of translating such findings into clinical applications. What are some of the challenges in moving from this genomic research to personalized medicine?
That's a good question. So one part is I would distinguish personalized medicine and target identification, right? So on one sense, what we've learned that could be applicable to all people, whether they contain an HNF1 alpha variant or not, is that if you created a drug that blocked HNF1 alpha, you would decrease lipids. That's actually done in phase 1 trials already, as I showed you. And what we might have to worry about is an increased risk of diabetes in those people, right? So we could monitor that people for that in a trial. And so what bad side effect to potentially look for. So that's for anybody being treated with this novel compound that targets HNF1 alpha. Now, from a personalized medicine aspect, the way I would frame the question is, what would I do if somebody came into my office and said, okay, I got my sequence, 23andMe or wherever, and I have these HNF1 alpha variants. now we know that people can have, we already knew that if they contained a loss of function variants, they had an increased risk for diabetes. Now we know that they could have a gain of function variant, and if they have that, they probably have a decreased risk of diabetes. But we should watch their cholesterol levels because they might be higher, and so that's a way that knowing their genetic information will help us to figure out what should we watch for in the future.
Thank you. So how can researchers effectively bridge the gap between basic genetic research and development of these targeted therapies or interventions?
Sorry, could you repeat that, Lucy?
How can researchers effectively bridge the gap between basic genetic research and the development of targeted therapies?
A big question. I think I can't give a comprehensive answer because I probably don't know enough, but I think one approach, at least to tackle that problem, is to use biobanks. And so here, before, genetic research used to be based on somebody comes to the doctor's office, they're sick, and that's why they get sequenced. And so people who are protected from disease would never be found that way because they're never coming to the doctor's office. And with biobanks, you have the opportunity to take whatever genetic research you did in the lab and directly intersect it with both people who are healthy and people who have conditions that you might be interested in studying. And so you can really look at that whole spectrum of who is at increased risk for disease and who is at decreased risk for disease, because everybody's donating their data, not just people who are coming to the doctor and getting sequenced because they're sick.
Thank you. Are there any genes such as HNF1 concerning the liver and pancreas?
I think if I understand that question well, are there any other genes like HNF1 alpha? I'm going to interpret it that way. And in fact, there's HNF4 alpha, which is actually activated by HNF1 alpha. And if you have deleterious mutations in that, you also get a different kind of MODY. What happens in the liver with regard to lipids, I don't think is known for that gene. But there's actually, I said HNF1 alpha was MODY 3. There are, I think at this point, about 11 or 13 MODY syndromes. So these are single genes which, when they're broken, cause diabetes, but they have different effects. Like one of them causes lack of development of the pancreas, PDX1. And so there's a whole host of these things.
Thank you. So another question came in, were there any patients with gain of function mutants also on a PCK9 inhibitor?
Without knowing specifically, I can guarantee you that the answer is no, because when these people donated this data and we collected it, the PCSK9 inhibitors were not available. But that's actually a great question because towards the personalized medicine angle, these people, if they have high cholesterol, a major reason for that would be HNF1 alpha activating PCSK9. So the PCSK9 inhibitor would be the perfect drug to treat them with because that's really precise and on mechanism.
Thank you. Have people looked for variants for longevity in Yamanaka factors?
Also a great question, but that's beyond my expertise, so I don't know.
Thank you. So how can advanced genetic techniques such as deep mutational scanning uncover hidden genetic variation and shed light on the diversity of human genomes?
I think the diversity is there, right? What we've learned is that from sequencing a lot of people is that every person on average, like every person in this room, this virtual room, has 300 protein coding variants that are unique to them. But most of us are, we don't have a Mendelian disease, we're not sick. So there is a lot going on biologically that buffers the effects of those mutations. where I want to take it is that these deep mutational scans can help you interpret these variants for genetics, and that's what I spent my whole talk on. But what this particular study is not about, but I think is a really exciting aspect, is using these deep mutational scans to do protein engineering. So if you take human insulin and you add 2 amino acids to it, you create a, a multi-billion-dollar drug that millions of people take because it lasts for a whole day. And now they have an insulin that's modified that lasts for a week. And now they have these GLP-1 agonists and these agonist peptides that are causing people on average to lose 20-40 pounds over 24 to 48 weeks. And it's really incredible. And this, we barely scratched the surface of this space. And so I think that's where, deep mutational scanning can really catalyze protein engineering and searching that space for actually peptides with novel properties that could work on people.
Thank you. So is there a role for optical pooled screening to be used to accelerate variant characterization?
Could I ask the person who asked the question, what do you mean by optical pooled screening?
While we wait for the person to answer that, the other question they had was, what are the advantages of using deep mutational scanning versus imaging readouts?
I think this probably relates to the optical pooled screening. So you know what? I'll while we're waiting for the clarification, I'll just interpret what I think is meant here. There, so Paul Blaney and others, Doug Fowler's group, have created some systems, which I think is what's being referred to as optical pooled screening, where you can assign a phenotype to a cell by roving a microscope, basically cell by cell, or building up barcodes in a cell by cell layer, and then ultimately imaging a whole field of cells that you've put variants in or done any perturbation to, and reading out something about them. So I think the deep mutational scanning is a method to introduce the variation, right? Another method to introduce variation would be CRISPR, where you're knocking out genes instead of introducing variants. And the imaging readouts or the optical pooled screening you would match that to the perturbation. So you whatever perturbation you want to use, then you could apply. In our case, we did FACS, and I think optical pooled screening is another method, a cutting edge method to read out on a cell by cell basis what is a particular variant or knockout doing. So I think they work synergistically together.
Thank you. So the abstract highlights, how certain genetic variants can seemingly have opposing effects on different physiological outcomes, such as protection from diabetes and increased secretion of atherogenic lipoproteins. How do geneticists and biologists approach understanding the concept of pyotrophy, where a single gene influences multiple seemingly unrelated traits?
That's also a good question. It's challenging, because I think we're in with all the buzz about AI and deep learning now, there's this idea that you just put in someone's genome sequence, and with enough of those, you'll just a machine will at some point be able to spit out what happens. And I don't think we're quite there yet, particularly in biomedicine and molecular medicine, because these things are, HNF1A was studied for over 20 years, and it was still unknown that there were gain of function variants. And so there's no, we have one of the first papers about that. And so if you put that into an AI algorithm, like 2 years ago, you get a completely different answer from now. So I think just that knowledge base is really still developing, and it's being developed in a very empiric way, right? We didn't know that there were going to be gain of function variants. We just did the experiments and then looked at what we found, and then and discovered them. There was no prediction. There was no, hey, they're going to be gain of function variants. We predict that through our knowledge of HNF1 alpha because it's been studied for so long and we know it so well. So we keep learning new things about even genes that have been studied for decades. And so I think we're just going to have to keep doing experiments and have an open mind, and not rely on predictive biology.
Mm-hmm. Thank you so much, Dr. Majithia. So I think with that, we're going to close the webinar. Thank you so much for your time today, and thank you so much for being a speaker for Twist. Before we close out, I have a quick reminder for everyone who's here. If you have 1 minute to spare, we really appreciate your feedback. If you could answer that survey. And thank you again, Dr. Amit Majithia, for such a wonderful presentation. And thank you, everyone, for coming.
Yeah, thank you all for attending, and thanks for the opportunity.
Take care.